Okay, so hello, it's 10:42. I hope your quiz went well. We're going to get on with our lecture today. Before we actually get into the next topic though, what I wanted to do was talk about the term assignment. So if you go to Moodle, the assignment should be up there now. There's a one-page or there's a handout, a sheet. I think it's maybe 3 pages, maybe it's 2 pages, I can't remember at the moment. It has all the instructions on there, and what you're going to do is you're going to choose a microbe. And so if you take a look at the list, I'll show you the list in a few minutes. There's a list of various diseases. Some of them are viral, some of them are bacterial, protists, a variety of different types of infections. So what I encourage you to do over the next few weeks as we start to look at bacteria and viruses and things like that is if you see something you like, then that might give you an idea which topic to choose. And then what you're going to do is you're going to make a web page or a fact sheet about it. And I'll show you some examples and what type of information we're looking for. Some of the information at this point may not make a lot of sense to you because we haven't talked about these things yet, like what is a gram positive versus a gram negative and things like that. But it will make sense soon over the next few weeks. So your web page or fact sheet is going to have some headings. And so you're going to use the following headings. You're going to use key points, classification, biology and mode of transmission, site of infection, pathology and disease symptoms, treatment, references, additional resources. So let me show you some examples here. So many of you may choose to do a website, and I'll show you, I found this blog about E. coli on the internet, so I'm just going to load that up here and show you what that looks like. So I think I just have to share, do a new screen share here. There we go. So here's a blog. This is something that was done professionally, so I just wanted to show you what it might look like, and it's worth it to take a look and see what these professional things kind of look like, give you an idea about what a good quality design of a website might look like. But this is a blog about E. coli, and so you can take a look at that. I will show you, I have also made a website. So I went to this website maker called Yola Site. You can make a free website from it. And I made this one here, so you can see MMI, medical microbiology, and there's a gram stain I did under a microscope. And you can see it says here, the research assignment is lots of fun, yay! So I built this page to show you how easy it is to build a page with Yola. I think this whole thing took me 10 to 15 minutes. Obviously, I don't have a lot of content on there, but the building of the website actually wasn't too difficult. There's some other tabs here. You see assignment details. It reminds you to use the following headings because I'm going to be looking for these things when I mark it. So make sure you do have the following sections. More information. You can make a fantastic web page. So I'll have the link for you for this on Moodle for you to check out. I have some other ones I'll show you. Here's some sample sites from some students. So here's a student who did leishmaniasis, and so you know, all these sample sites from students, these students did well, you know, not perfect marks, but you can get an idea of what it looks like. You can see she has the key points, maybe a little too detailed in the key points. That's okay. She had a video. It doesn't look like it's active anymore. This was from a couple of years ago. We can go on additional information. You can see she has the classification, a nice map. Notice the map has a reference, okay? If we're borrowing somebody else's images, we've got to make sure we reference it appropriately. So in this case here, it's fine if you just put the URL link. She's got a little bit about the biology and the life cycle and the pathology and the disease symptoms. Lots of nice pictures. It was very well researched, this particular project. And lastly, we've got resources. So I'm not asking you to reference this like you would do an essay, so you're not using like a PA format or Chicago style or anything like that. What I'm looking for are some resources, okay? I understand that you're going to get the information from all sorts of sources, web, your textbook, etc. What I want is some resources at the end for someone who is looking for more information. So, for example, your textbook could be referenced. At least one of your sources should be a non web resource anyway. So use your textbook or a journal article or something like that. If you do use web links, you can see here, she has some web links. Don't just put the URL in your reference list. Make sure that you do put the author and title. So maybe the author is the World Health Organization, the title is leishmaniasis, and then the URL. So they have an idea what they're looking at. There are many good, reputable resources out there. You can use the CDC, Public Health Agency of Canada. Do not use something like Wikipedia. You may use that as a starting point, but do not reference Wikipedia, please. I'll show you another one. Here's one on malaria. This one's a little different, but you can see it has the same things. It has key points, lots of information, some nice pictures, maybe a little fuzzy on that picture on the right. So try to make sure your images are nice and clear. That's important. And your font sizes are a decent size. I want to make sure I can read it. Sometimes people want to use really tiny fonts or they want to use really bad colors. So make sure your colors are easily readable. Like don't use a yellow font on an orange background or something crazy like that. And references. So you can check these out and see what you think, and maybe it'll inspire you for some ideas. Here's one on chicken pox. So there's chicken pox, and you can see there's a cute little picture, and same idea. This one here is very nice and clean. Okay, I gotta click down here for some more information. So relatively simple and straightforward. It is going to take you a little bit of time to build this and collect the information, but hopefully you get the idea there. I'm going to go back to my PowerPoint here. Let's just make sure I'm sharing that properly. Okay. So you may not choose to do a website, and that's fine. You may want to choose to do a fact sheet or a pamphlet instead. It should be 2 pages, no more, and that in some ways might be a little bit more difficult because you really are tight for space. But here's a sample. This is actually a professional done fact sheet on MRSA. You can see what it looks like, and it's very nice. Has some clean pictures and some other information. I'll show you some student ones. So here's a student one on tuberculosis. And this one here, I think it was designed to fold into a 3-way pamphlet, if I remember correctly. I just have it scanned flat. You can see the person has, again, some nice pictures and some information there. Some bullet points, maybe they could be spaced out a little nicer. You know, presentation is part of your grade, so do make sure it looks nice. Like I said, clear pictures, you know, consistency in terms of your font size, good spelling, all those things do count. Here's the back side. You can see the information. This one's maybe a little bit sparse of information, but again, you can only add so much on the pamphlet. So you really have to be very specific and make sure that you do add the right type of information. Here's another example. This one is on syphilis. Sorry, it looks like the scan didn't turn out as sharp as I'd hoped, but actually this is a good example of what not to do in terms of the font. The pamphlet was good, but you can see the green background with that, you know, kind of greenish font. Maybe not the best choice on that particular pamphlet. But this one here, it looks like it folds in on itself. So here's another one. This one is on the beef tapeworm, and you can see I put a couple notes on there. But other than that, this was a pretty decent back page. It looks like he or she did not reference their images, and maybe could use 1 or 2 more. But other than that, the information was pretty good. So here's the topic list for this year. I have a different topic list every year. If you do have something that is not on the list, you can run it by me and I may grant you permission. I do not grant every topic every year. Try to cut down on plagiarism. Some of these things you may have heard of, and some of these things you may have no idea what they are. I encourage you over the next couple of weeks, spend a little bit of time on Google and learn, a little maybe about these, and you'll find something you like. It may turn out that you can't find a lot of information on something as well. So maybe you're doing Mycobacterium avium complex infection, and it's just seems a little too complex for you. Then choose something else. I just realized I had chickenpox on there. I may have to take chickenpox off the list because the sample one I gave you was on chickenpox. So please ask me if you're interested in that. Okay, I think I've got a couple of questions here. Someone says, can we choose any microbe, not just the ones on this table? I do prefer that you choose one on this table. If you do have another idea, run it by me first, and I will let you know whether it's suitable or not. There's some diseases that are just not suitable, like there's multiple causes, and it just makes it very complicated for this kind of thing. Or things like syphilis is just a very hard one to do in 2 pages. It's just a very complex disease. Okay, here's the grading rubric for this. So you get an idea of what I'm looking for when I grade this. I'm looking for, obviously the right information in the various sections. It's very important that you do read the instruction sheet that I have on Moodle. Okay, a lot of people do lose marks for not being complete in some areas. For example, the section that says treatment or immune system response, it does say in the notes if there are preventative measures, right? And a lot of people forget to mention, for example, some diseases there is a vaccine for. That is kind of important. Preventative measure. It's not a treatment, but it is a preventative measure. So do make sure that you do look at the details on the instruction sheet. That's very, very important. Key points should summarize the other sections. So some people go a little too crazy with the key points and they add too much information. Sometimes people do not add enough. Okay, so do make sure that you do follow that. And presentation. So like I said before, good spelling and organization, a nice, clean project that's good to look at is going to get you better marks there. So this usually helps people's marks a little bit. I think the average on this is typically around 80%. You do have an opportunity to do well on this. Some people don't do well. Some people try to do this all in the last day or 2 before it's due and don't do a very good job either. So I encourage you to work on this early. You have lots of time. I can't remember the due date off the top of my head. It was on that first page. It's in November. So like I said, hopefully next couple of weeks gives you an idea of what you may want to actually look at in your project, and then give you lots of time to read about it and do a good job on it. And I hope it's lots of fun. I think students usually find this to be a fun project. I'm sure you write essays in other classes, and I certainly don't need to write one more, and maybe learn some different skills and all that. So I've had some questions about, I don't know how to design a web page or anything like that. Take a look at those links I have in the sheet. There's lots of different sources that on how to do it. I mentioned Yola's site. There's one called Wix, I think. There's there's ShareSpace. Some of them have free. Some of them have paid models. I think I had somebody last year who used a paid website and they didn't want to pay for it, and so they just sent me their password, and I was able to log in and take a look at their website and those kind of things. As for if you do a pamphlet or a fact sheet, you can try PowerPoint or Word. There's publishing softwares. I'm not going to tell you what software to use. I can try to add some assistance for what I do know, but try a few things and see what works. I encourage you to do that. So do not forget, by the time you hand in your assignment to do the plagiarism certificate. A number of people have already emailed it to me, and that's fine. There's no rush. You don't need to hand it in until the assignment is due. But just a reminder, I'll remind you again when the assignment is due to hand it in, because I will not be grading your assignment until I get the certificate. Okay, so I think that is it for the assignment for now, unless there are any questions. We will go back to our lecture. So where is the lecture? There it is. Am I sharing the right screen? I believe I am. Just make sure that goes up. Okay, there we go. Okay, so today we're going to start talking about bacteria. We will not be finishing this all in one lecture. This will be 2 lectures, so today and hopefully we'll be finished on Thursday. Looks like I have a question here. So this person says, was this the only quiz we have all semester? The answer to that is yes. We have 2 midterms. So the quiz is worth 5% and the midterms are worth 20% each. And you'll have to check the course outline for the dates. I don't have them off the top of my head, but I think the first midterm is about 3 or 4 weeks from now. So check those dates. It's early October anyway. October 1st, somebody just wrote. So that is coming soon. Okay, so let's talk about bacteria. So this goes back to last week. We were talking about eukaryotic and prokaryotic cells. And so if you remember, the big difference was the nucleus. And you could probably put size in there as a major difference, is that eukaryotic cells usually are much, much larger. I think I have another slide here, yes. And this was the other slide I showed you when we talked about some of the cell components. So what I want to do now is I want to go back to these notes we made. So I told you we were going to make some more notes. So I made this table last week. We were looking at differences between prokaryotics and eukaryotics. And there's another question here. So someone is asking, is there going to be a way to see the quiz questions we just did if you wanted to see your mistake? And the answer to that is I think I have it programmed so that you can go in and review it, but I'm not entirely sure whether that worked or not. So it looks like someone's confirming that you can. All right, so back to prokaryotes and eukaryotes. We made this table here last week, so we were comparing them, sizes. Prokaryotes are smaller, eukaryotes are bigger. No membrane-bound nucleus or organelles for prokaryotes, and we talked a little bit about cell walls. So bacteria have cell walls, and eukaryotes, not in animals. Fungi have cell walls, and protists, some of them have cell walls. And then I gave some examples. So today I want to expand this table here a little bit, and some of these things I'm going to write down here. We haven't quite talked about yet, but I thought I'd fill in this table for starters, just to give you a bit more, to flesh it out and give you a bit more information. So first one on the list there you can see is DNA structure. And again, you know, we can, in biology, start most of our sentences with usually. So prokaryotes usually have a single circular chromosome, whereas eukaryotes usually have multiple linear chromosomes. So you probably know that humans have 46 chromosomes and they're linear. Most prokaryotes have 1 and it's circular. They have smaller genomes, prokaryotes. I guess I can put larger genomes. Quite a bit larger, like massively larger. And I had one more note, is that in prokaryotes, DNA is in what's called the nucleoid region. So we'll talk about what that is in a couple of minutes here. Eukaryotes, the DNA is found in a membrane-bound nucleus. Okay, so that's the first part, comparing DNA structure. We move down. Next part is ribosomes. So it turns out that prokaryotes have smaller ribosomes. And the size, actually, there's a size unit called S. And we'll talk about that in a couple of minutes as well. And of course, eukaryotes have larger ones, which are ADS. Someone's asked me to scroll up just for 2 more seconds. There you go. I'll just leave that for a second. This is the huge advantage of not having the whiteboard. I like to have my PowerPoint and I like to have the whiteboard all on simultaneously so that people can see 2 things at once. All right, I'm going to scroll down now. You can always take a look at the lecture videos as well if you do miss something. All right, ribosomes, like I said, prokaryotes have smaller and eukaryotes have larger ribosomes. Reproduction. So prokaryotes do something called binary fission. We'll talk about what that is as well. Eukaryotes, we're talking about mitosis and meiosis. Prokaryotes are always asexual reproduction. Eukaryotes can have, oh, I guess I can't spell again. 1S, there we go. Eukaryotes can be asexual and sexual reproduction. It just depends on the species. And some, in fact, like fungi, some fungi can do both. One other point about reproduction is that bacteria can exchange DNA by a process called conjugation, and we'll talk about what that is as well. So something else to look forward to. So somebody has a question that says, are ribosomes considered to be organelles? That's a hard question to answer because some people call ribosomes organelles, some people don't. Usually the definition of organelle is something that is membrane bound, and ribosomes in both prokaryotes and eukaryotes are not membrane bound. So they're not necessarily falling into that category as an organelle. So I more or less just call them, cell structures or cell components. I usually don't refer them to them as organelles, but some people do. Okay, motility. So that means moving around or swimming. So bacteria, some of them have flagella. And these flagella rotate. So rotating, if you think about like a fan or a boat propeller or something like that, that's how they move. In eukaryotes, they have things called flagella, but the flagella are completely different structures and they're more like whips. So I'm calling whip-like flagella. And they also have things like called cilia, which we're going to talk about. That will be next week. We'll talk about those structures in particular. And they also have something called amoeboid movement. Again, that'll be more for next week. So a bit more on that later. All right, so the last category is here endospores. Again, you don't know what endospores are yet, but we will talk about them. Maybe we'll get to it today. And those are found in some gram-positive bacteria. So again, we will talk about what gram-positives are as well. Gram-positive bacteria. And then eukaryotes, no. But some protists form something called cysts. Okay, so I know that was a big information dump for everybody. And there's a lot of words and concepts in there that we haven't covered yet, but I thought it was a good time to just introduce some of these things, particularly when we're starting to think about the differences between the different types of pathogens, the eukaryotes and prokaryotes. And so I figured it was a good time just kind of introduce some of these concepts, and we will get through there. We will, there'll be lots of repetition, which I hope will help you learn what these things are. Okay, I'm just going to give you about 5 more seconds here in case anyone else needs to finish up, and then I'm going to go back to the PowerPoint. Okay, here we go, back to the PowerPoint. Okay, so that's enough for eukaryotes right now. We're going to talk about prokaryotes today. So back to this diagram, you may remember we have the 2 types of prokaryotes, the archaea and the bacteria. In this class, we were focusing 100% on the bacteria because you can see that big note, the archaea have no known pathogens. As far as we know, they don't seem to make us sick. Who knows, maybe someone will find an exception someday. So what can we say about prokaryotes? They seem to be everywhere. Absolutely everywhere we look. It's really incredible on this planet. They've been found in nuclear reactors. People find them in these mines that are miles deep. They seem to be everywhere. They're very abundant. Handful of soil will contain 10,000 species. They just seem to be absolutely everywhere. These include the bacteria and the archaea, probably there's more biological mass from prokaryotes than anything else on the planet, but we're not really sure. It's very hard to measure all that stuff that's going underground and in the oceans. Let's talk about the archaea just for a minute. Sometimes textbooks will say archaea are extremophiles, and what I mean by that is organisms that prefer extreme environments. So you can see in this case here, we have a hot spring found in some national park. And if you go there and you stick a microscope slide in there, you've got this water that's like 90 degrees Celsius. And you stick that microscope slide in, you collect it a couple of days later, and there's something that looks like bacteria on there. But they're not bacteria, they're archaea. They're biochemically different, structurally different, and they're determined to be very different by biologists. But they're not all extremophiles. Many of them are found exactly other places where bacteria are found in soil, in your gut. Here's a little cartoon talking about where they might be found in your gut. You can see this girl's talking, and then she has a little bit of flatulence. And it says, don't be embarrassed. It's not your gas, it's your archaea. So yeah, some of these archaea are methanogens. They're making methane, which are leading to some embarrassing moments, of course. Anyway, like I said, there are no known pathogens, so this will be it. I probably won't talk about them again. In fact, when I talk about prokaryotes from now on, I will be talking exclusively about bacteria. It's not that these things aren't interesting, they just, medical science, they don't have any relevance at the moment, unfortunately. Okay, so let's talk about the bacteria. What do they do? These are kind of repeats of things that I said last week. Many of them are symbiotes with us. So E. coli lives in our gut, and it eats our food and provides us with some vitamins. Some of them are very nasty pathogens. So there's the one that causes the plague. These are the ones we're going to be focusing on in this course, of course, many of the pathogens. Many are photosynthetic and important parts of the food chain. Many are important in nutrient cycling, and the list goes on and on and on. We make all sorts of food from bacteria. They are helping animals to digest grass and cellulose. They do all sorts of cool things. Okay, so bacteria's pathogens. We've already talked about anthrax a little bit. So I want to introduce you a little bit more to anthrax. There's a picture of anthrax. I can't remember if I showed you what it looks like before. It's sort of almost spaghetti-like, and you can see it stains purple under a gram stain. And so what can we say a bit more about anthrax? Anthrax kills many animals. I think we mentioned that before. Historically, it used to be a huge problem with livestock, particularly livestock that dig around in the ground. So bison, they eat grass and they've got big noses. So sometimes they inhale endospores from the anthrax from the soil, assuming it's in the soil. It's not found in soil everywhere. And so this is showing a bison that was killed by anthrax in Alberta a number of years ago. There was a big outbreak in one of the herds. And today, livestock isn't as much of an issue because we have antibiotics and we have vaccines and things like that. But back in the day, it used to be a very significant issue. There's really two types of anthrax that people can get. One is inhalation anthrax. That's pretty rare in humans. We're not usually snuffling around in the soil. But if it gets in the lungs, that can be a pretty serious disease and life-threatening. Most humans are getting cutaneous anthrax, which is in the skin. So anthrax is an organism, it has toxins in it. The toxins kill the skin. That makes the skin something called necrotic, turns black, and you end up with these black blisters. And that's usually relatively treatable by antibiotics. So there are many, many pathogens that are caused by, or that are bacterial in nature. Here's a list of the few that we're going to talk about. There's a bunch more. And if you take a look at your notes, at the end of each set of notes, I have a list of featured pathogens. So hopefully by the end of the semester, we'll get through. You'll see it's going to be a pretty long list by the end. Some of these you may be familiar with. Some of these you may have had. Some of them you wish you'll never get. But bacterial infections are a reality to us, and so they're very important. In fact, about 85% of the infectious diseases known are bacterial in nature. So they're a very, very important part of this course. Okay, so for each unit, I have a case study or two, and the whole idea behind the case studies is to give you an idea, a little bit of an idea of how some of the information we're learning is relevant to case history of patients. Some of the case studies you'll be able to answer pretty easily. Others will be a little harder. I'll have to kind of help you out along through. But I will have case studies as a part of all the midterms as well. And in that case, well, when we get closer to the first midterm, I'll give you some more examples. So here's an example here. We've got a 15-year-old male, and he's got some symptoms. And notice one of the symptoms is a pus-like drainage in his underwear on the tip of his penis. So this is probably a sexually transmitted disease of some sort. That would be kind of a hint there. So one of the first things they do is get a sexual history, and yes, there has been a history, it looks like, of 5 partners in the past 6 months. And apparently none of them have any sexually transmitted disease, but we should probably get this kid tested anyway. So it says here, here's the results. He got a urine test, and the urine culture was negative. But he did have a discharge from his penis, and they did a gram stain on the discharge. And you can see the note here. It says they have gram-negative diplococci. So probably for most of you at this point, that means nothing. But by the end of this lecture, this is going to mean a lot more. I'll tell you the answer right now. This is Neisseria gonorrhea. And the reason I know that is because there's not a lot of organisms that fit that category, gram-negative diplococci. But we're going to cover what that is. We'll come back to this case study in a few minutes once we get a chance to talk about what these things mean, gram-negative and diplococci. So we're going to start looking at bacterial anatomy and the structures of bacteria. Many of these are really important for virulence. That means the ability to cause disease. And many of these things are also important because they are important targets for our drugs, our antibiotics. So I showed you this picture or something similar before, a picture of a bacterial cell, and you can see a bunch of features on here, and we're going to basically go through these one by one. Some of these features you've probably heard of, and others are probably new to you. And as we go along, we'll make a few notes to go along with that as well. Okay, so first thing to talk about is the genome. So I mentioned already that the genome in bacteria is usually circular. So you can think of kind of like a big hula hoop, but rather than a big open hula hoop, it's more like twisted and tangled, although not tangled in a way that's causing knots, more of an organized way, but it's sort of like an elastic band that's squished up somehow. You can see this particular cell here is undergoing binary fission, so it's dividing, and it has two genomes. So because its DNA is replicated, so it's starting to divide, and each other cell is going to get one copy of the genome. And the genome is in something called the nucleoid region. So what is a nucleoid? It's not a nucleus, okay? It's not membrane bound. The reason why it's called the nucleoid is because it is visible on electron microscope. So it was named by people who were doing electron microscopy. They see a dark stained structure and they gave it a name. Hey, there's the nucleoid. I don't know why they didn't just say, hey, there's the chromosome, but this is the terminology that is stuck. I have one more thing on this slide, and then I'm going to make a couple of notes. Sometimes bacteria have little mini genomes, and these are called plasmids. So the normal chromosome, if you think of something like E. coli, that's about 4,000, 5,000 genes. A plasmid might have 3, 5, maybe 15 genes. So it really is like a mini genome. So what's the point of these plasmids here? Well, it turns out plasmids are something that organisms can trade, kind of like trading cards. It's like, hey, I've got a gene or two here you might like. And they do that. And often plasmids, unfortunately, medically have virulence factors in them. So let me show you a little table, and then I'll make some notes on these things. So here's a little table that says some traits were coded on plasmids. So take a look at that first one, antibiotic resistance. That's the one that we worry about the most in hospitals. You can have a plasmid with resistance to a drug, and it can be spread between different bacteria. That's kind of scary. Some other things we haven't talked about yet, but plasmids can code synthesis of pili or pilus. Toxin production. The plasmids can have toxins on them, and a whole bunch of other things. Okay, I'm just going to go back to this slide here, and we're going to make a few notes on these things. So I started a Word document as well. Okay, here we go. So here's my new Word document. This is usually what I do on the whiteboard again. And you can see I have a table I'm making. So I've got the structure, I've got the composition in that what is it made of, and then I'm going to write a little bit about the function here. Okay, so the nucleoid, of course, is chromosome. And what is a chromosome? A chromosome is DNA, and it also has some associated proteins. But you don't need to worry about those associated proteins in this class. Mostly they're just organizing the genome and making sure it doesn't get tangled. So what's the function of the nucleoid? This is where the genome is found, contains the instructions for the cell. So hopefully that one's not too hard. What's a plasmid? A plasmid is circular DNA. And like I said, you can think of it as a mini chromosome, very mini. So a plasmid contains a small number. So it could be 1 to 15 is typical. Actually, 1 to 5 is more typical, but I'll put 15 because that's some do have that many. There's a small number of extra genes. And I'll put another note, may contain genes for antibiotic resistance. That's the one that we're worried about the most. But they do carry, like I said, other virulence factors that we're concerned about. So plasmids are something that you hear about in the scientific literature a lot. Okay, I'm just going to give you a couple more seconds on that. We will come back to this table. If you do miss something, I'll come back to it once we look at the next structure. I'm going to switch back to the PowerPoint now. Okay, I already saw that slide. Okay, ribosomes. As I mentioned, ribosomes in prokaryotes and eukaryotes are a little different. It turns out the ribosome in the prokaryotic is a little bit smaller. You can see that number, I have 70S. You're probably wondering what the S stands for. The S stands for a guy named Svedberg. Svedberg was a Swedish guy. He was studying ribosomes, and so they named the unit after him. It's basically a measurement of size. Smaller number is a smaller size. If you take a look at a ribosome, it can be broken down into two subunits. You've got a large subunit and a small subunit, and the composition in the prokaryotes and the eukaryotes is actually different. And so this is actually relevant because ribosomes are actually a target of some antibiotics such as tetracycline. So let me go back to our note here, and I don't know if we really need a note for ribosomes, but we'll put it there anyway since we already did a note last week. So what are ribosomes made of? Ribosomes are made of ribosomal RNA, and they're also made out of proteins. This is the site of translation, which is protein synthesis. Okay. Okay, so I mentioned the word binary fission before, so you're probably familiar with mitosis and meiosis. You've got your prophase, metaphase, anaphase, telophase, all those fun things. So what is binary fission? Well, if you think about mitosis and you've got all those special things going on, you've got those spindle fibers invading the nuclear space, and you've got everything lining up at metaphase, and you've got all the separation at anaphase. It turns out when you have one chromosome, you don't need to do all that fancy stuff. It's actually very simple. So binary fission, binary means two, of course. Fission means splitting, right? So if you think about nuclear fission, you're splitting of the atom. So it just means splitting in two. And it's not quite that simple, but it's not nearly as complex as mitosis. So mitosis in a human cell takes about 16 to 20 hours. Binary fission in E. coli, under ideal conditions, can actually take 20 to 30 minutes. Not all bacterial cells are as fast as E. coli, but it's very, very simple. So take a look at this image on the left, and you can see we have one cell at the top. And I thought, see if I'm just going to grab my pointer pen here. So I got one cell at the top, excuse me. Just want to point something out with this chromosome right there. Is that the chromosome is actually attached to the plasma membrane. So you can see the first step here is the replication. Interesting, they're calling it duplication, but the term we usually use is replication. And so the chromosome is attached to the nuclear membrane. You can see that cell is starting to grow and it stretches out. So it turns out that this chromosome here on the left, it stretches out to the left, and this chromosome on the right stretches out to the right. And so as the cell stretches out, the chromosomes are separated. And then basically at the end of that, a septum forms. That's the two cells are dividing into two, and you end up with your two daughter cells. So that's binary fission. So mostly the thing I want you to know about binary fission is the definition. Okay, that is, it's important to know what it is, as it may be mentioned, so it's important for you to know what it is. Okay, so how do we classify organisms? We talked about this a little bit already, as we talked about taxonomy, how we take a look at shapes and sizes of organisms, and do they have hair and certain types of teeth and those kind of things. For bacteria, we're looking often at things like shape in response to stains, and that's historically used a lot. Also biochemical characteristics such as can they digest lactose, can they grow aerobically or anaerobically, those kind of things. More recently, we're looking at serological characterization, so that's looking at how the immune system responds, and also, as mentioned, we're also looking at what their genome sequences look like. And we're typically looking at ribosomal RNA sequences because ribosomes are found in all organisms. So a little bit more on those things, that serological and DNA stuff. That's kind of more after the first midterm. It's material for the second midterm. Okay, so some other words for you to learn are morphology. So morphology means shape, and So here's some shapes to learn. We've already learned this shape here, coccus. Remember staphylococcus. Coccus means spherical or round shape. Here's another one to learn is bacillus. So bacillus means the rod shape. And vibrio means a bent rod. And then lastly, sporillum. Sporillum means a spiral shape. Sometimes we use words in front of the shape to give a bit more information. So diplo. So diplo means two, right? So diplococci. So it means two spheres tacked together. So you can see there's some pictures there of some diplococci. So Neisseria gonorrhea, I mentioned before, falls into that category. And sometimes streptococcus will actually look like diplococci as well. But they're gram-positive, so they'll stay in a different color. So more on what that means in a minute. Strep. So you may have heard of strep throat. Strep means a chain. So you can see streptococci are forming chains of cocci. Cocci, of course, meaning spheres. So you can see there's a couple of streptococci there. Streptococci pyogenes, that's the one that causes strep throat. Streptococcus pneumonia, that's an organism that causes pneumonia. So if you're lucky, so you can learn something from the name of the organism. Neisseria gonorrhea. Anybody's guess what that causes? It's what you don't want. Pyogenes, I think that's from the word meaning fever, pyo. So people will often get a fever from strep infections. And I have to double check that one by looking that one up, but I think that's where the name comes from. I can't seem to advance my slide. Oh, here we go. All right, then there's staphylococci. So we talked about staph already. Staph is a cluster, and you can see there's Staphylococcus aureus there. We also talked about Staphylococcus epidermidis, and there's a gram stain, and I think maybe I took that gram stain. You can see they're nice clustery spheres there, make a nice, very easy to identify. And then there's bacillus, right? So sometimes you do see people saying diplobacillus. Sometimes you see people using other terms with bacillus, but often you just see bacillus alone. Bacilli is plural, bacillus is one. So if you see both of those terms, you see bacillus is in the name of a lot of organisms. So bacillus anthracis for anthrax, we talked about that one already. There are unfortunately a lot of organisms that fall into the bacillus category, so you can only use it for the genus of one group and not the others. So E. coli is bacillus as well, and as are many others. Okay, so you should know these terms here. There are other shapes and terms, but these are the ones I want you to know, partly because a lot of them end up in the names of organisms that we're going to be talking about. So review this slide in a little bit over the next couple of weeks, and hopefully that will help you learning some of the names of some of the bacterial organisms that we're going to talk about. Hopefully, just due to repetition, these things are going to pop up again and again and again. Bacillus, Staphylococcus, Streptococcus, et cetera, et cetera. These things are going to pop up again and again. Hopefully, through repetition, you'll learn it. Here's a few other names. I just found some of these organisms. Streptococcus, I guess we mentioned that one already. But take a look at this one. I don't even know what the last two organisms are. Canobacillus. This one I just found on the internet. I haven't even looked it up yet, but I know already from the name, it is a rod shape. So it's going to look something like this. I don't know what color it's going to stain yet. I don't know anything about it. Aquasporillium. Bengal. I don't know if that has something to do with Bengal tigers or what, but I know it's going to have a bit of a spiral shape to it. So it might look something like this. Okay, so let's talk about a few other bacterial structures, and I want to get back. I'm hoping to get back to that case study today. I want to talk about peptidoglycan. So we were talking about peptidoglycan last week. See, there's a question here. It says here, is there anything that the color tells us? And yeah, so I'm going to get into the colors here now in a minute. I'm going to talk about gram positive and gram negative and the purple and the pink stuff. So we'll get to that quite shortly. So let's talk about peptidoglycan. That's a big word. I know a lot of people are intimidated by it. But if you take a look at the word peptido, that means peptide, right? So amino acids. And glycan. Glycan means carbohydrate. Anytime you see carbohydrate or glycan, so I'm just going to write that down here, carb, and this means amino acid. Sorry, I don't have my pen today, so I'm just doing this with my mouse, and it's certainly a lot slower to write with my mouse than with pen, and not as neat. So what is this thing, peptidoglycan? If you take a look down here below, we have N-acetylglucosamine or NAG. We have N-acetylglucosamine or NAN, or N-acetylmuramic acid or NAN. I don't expect you to learn those words. I don't even expect you to learn NAM or NAG at all. Just know these are carbohydrates, and structurally, if you're interested in biochemistry, they look a lot like glucose. They really are just decorated glucose. And where's my mouse? Oh, there it is. Sorry about that. If you look at this here, you know, they've got all these weird functional groups on here, and that just makes them not glucose, gives them some fancy names. So these carbohydrates, if you take a look on the right, they're found in strands of carbohydrates. So you've got all these strands, these fibers, and they're linked by these amino acid chains. So what you end up, I'm going to kind of draw you a little picture. I know I don't have a lot of space, but here's my little carbohydrate strands, right? So this is how you make a cell wall. You put a bunch of carbohydrate strands together, and then you kind of link them together with these peptides. What you're doing is you're kind of making a mesh-like structure. And that's really what peptidoglycan is. It's a very strong structure found in the bacterial cell wall, and it's just holding those cells together. So it turns out that there's actually two types of bacterial cell walls, and this is found out from a certain type of stain called a Gram stain. We're going to be talking about Gram stains here and in a couple other units. And it turns out that this divides most bacterial cells into two categories, the gram positive and the gram negative. So let's take a look at what the difference is between these two different types of cells. Okay, so take a look here first on the left. We've got the gram positive. Gram positive, if you take a look here, it has this very thick layer of peptidoglycan, and then over here it has a plasma membrane. So that's probably what you think of in terms of traditional type of cell wall. You've got a plasma membrane, and outside you've got a thick cell wall. And if you take over here on the right at the gram negatives, you've got a plasma membrane here. So here's my plasma membrane, PM. We've got a very thin peptidoglycan layer, so we'll call that PG. And then we actually have a second membrane called an outer membrane. So it's kind of a little bit more like a cell wall sandwich. So it's a bit of a different structure. And it turns out these things, when we stain them using the Gram stain, they turn out different colors. So why am I mentioning the Gram stain? The Gram stain is probably one of the most important stains for bacteria, historically and even in modern clinical times, because it's a very easy way to diagnose some organisms very quickly. So what happens with the Gram stain? You can see over there on the left, my slide got ahead of itself. It's showing its stains with crystal violet. So violet, of course, is purple, and so Gram positive cells stain purple. Gram negative cells do not stain purple. And we're going to talk about the procedure on this in a little bit of a later unit. But basically, you've got that outer membrane. It protects the cell, and the purple just does not permeate very well. And so we use a second stain that's called a safranin red, which is kind of a reddish, sometimes pinkish color. And so they stain pink instead. So like I said, we'll take a look at the exact procedure in more detail in a later unit. I'll show you some pictures here. So we've already looked at anthrax. It's purple, gram positive. And there's E. coli on the right. E. coli is staining pink or red, and it is gram negative. So we're going to be talking a lot about these different organisms, and there are biological differences between the gram-positives and the gram-negatives, and we'll be highlighting some of those and why it's important sometimes to know whether an organism is gram-negative or gram-positive. So more on that in the coming units. So I want to point out a couple of special features about the gram-negative cell wall. So there it is. Like I said, think of it as kind of like a little bit of a cell wall sandwich. You've got two membranes, and there's two things I want to point out. One is this area called the periplasm. So we have our cytoplasm, and now we have a little bit of a tiny, tiny space just between the two membranes, and it actually contains enzymes. And some of those enzymes are involved in building the outer membrane and building the cell wall. But it also often contains enzymes that are helping these organisms be more antibiotic resistant. It turns out that gram-negative organisms are more naturally resistant to drugs than gram-positives, which is important clinically. The other thing I want to point out is that the outer membrane has a very unique lipid in it called LPS, or lipopolysaccharide. Why is this unique and why is it important? It turns out that this is something called an endotoxin. So we're going to be talking about endotoxins way at the end of the course when we get to the immune system. But this is something that our body does not like. So if you get bacteria in your blood, that's not a good thing. And one of the most dangerous features about getting a bacteria in your blood are these lipopolysaccharides. So if the bacteria cell breaks up, maybe due to an immune reaction, and these lipopolysaccharides end up in your blood, your immune system recognizes them as not human, not self. And you can have a significant immune response, which can be in some cases fatal, not always. In some cases, just leads to sweats and low blood pressure and things like that. But it's not good to have lipopolysaccharides in your blood. Okay, so back to this case study. And I'm not going to go much further today than this. I know I've probably filled your brains, and maybe some of your brains are tired from the test, but I want to go just a little bit further after this, and that'll be it. It'll probably be done early today. So back to the case study, we mentioned that we saw a gram-negative diplococci. So the question says, which organism is this? Neisseria gonorrhea. So it turns out that there's actually very few organisms that fit that description of being a gram-negative diplococci. At least very few clinically significant organisms. There are a few of them out there. The other clue is that it's a sexually transmitted infection. There is another organism, it's called Neisseria meningitidis, causes meningitis, and that's not usually found in a discharge from someone's urethra. And so in this case, the response is the person has gonorrhea, and they're going to be put on antibiotics, probably prescribed right away. Doesn't mean that the organism is not resistant to those antibiotics, because you do find that with gonorrhea from time to time, but usually that's the first course, is going straight to the antibiotics after this gram stain. So diagnosis directly after the gram stain is pretty common with some organisms. Okay, so I did want to mention that gram-negatives and gram-positives, I mentioned that was most bacteria fit in those categories. There is one other important group I want to mention, and these are called the mycobacteria. And why are they called mycobacteria is because they have something called mycolic acid. So let me show you, I've got a picture here. This is what a mycobacterial cell wall looks like. You can see it has these mycolic acids. So what do we care about those mycolic acids? Well, it turns out they're really, really waxy substance. Kind of like, almost like a birthday candle, maybe a little softer like that. So it turns out that these are, have a waxy surface on the outside, and if you try to gram stain them, they don't take up the gram stain. So we have a special stain process for this. It's called the acid-fast stain. We'll talk about acid-fast stains as well later on after the midterm. And this stain here is a blue and pink stain. So you can see there's Mycobacterium tuberculosis being stained there. So it's a bacillus, it's a rod. So you probably know Mycobacterium tuberculosis causes tuberculosis. It's in the name. Mycobacterium tells me it has that mycolic acid, that waxy layer. So this is a little bit of a different type of cell wall. Like I said, you can see from the picture, it's got a similar structure to a gram-positive, and then it's got some extra layers of stuff on the outside that makes it more protective and harder for our bodies to, our immune systems to deal with, which is why people have chronic infections with tuberculosis. Okay, so I'm back to this picture. I just want to go and I guess make a note here about peptidoglycan. So I guess I got to share that slide, don't I? Share. Okay, what happened to my mouse here? I got to figure out. My pointer's disappeared somehow. Sorry, just give me a second here. Okay, I'm going to go back to my Word document and write a note on peptidoglycan, and that's where we're going to finish today. So what is peptidoglycan made out of? It's made out of carbohydrate and amino acids. And what is the function of peptidoglycan? It provides structural support as the component of the cell wall. Also provides cell shape. Okay, so I think that is where I want to finish for today. Please let me know if you have any questions. If you have any follow-up from the quiz, you can fire me off an email as well. I'm going to stop the Zoom meeting in a minute if there's no questions. I don't think I have anything else to say other than we will hopefully finish this topic on Thursday. Moodle should have marked your quiz right away and put your grades up in there. If not, I will make sure the grades are inputted in the next day or two. Anyway,